So good afternoon, everyone. Welcome to the post-lunch session of the second day event. That is Atal Academy sponsored FDP on recent advances in biosimilars development. Now we have a team from Ingenuity Biosciences headed by Dr. Ravi Krovidi. Is going to deliver a talk in this session. I will give a brief introduction about him. He has over 20 years of experience in biologics and biological products, and currently he is serving as a CEO of Ingenuity Biosciences Private Limited, Ahmedabad, and is also heading Biosimilars business unit at Vida Clinical Research. Earlier, he served as a head Center for Advanced Protein Studies in Syngene Internationals, and he has also worked in various national and international organizations. And he has received his PhD from Max Planck Institute of Immunobiology in Germany, and he has worked in the area of quantitative proteomics, LCMS, and transcriptomic analysis. And he has also presented his scientific work in various leading international scientific conferences. With this brief introduction, I'm handing over this to Dr. Ravi Krovidi and team for the talk on advancing biosimilars from development stage to clinical evaluation, overview of key technological advancements and regulatory insights. Over to you, Dr. Ravi.
Sure. Thank you so much, Dr. Somsundram. It's thanks for the invitation and the kind introduction. Good afternoon, everyone. I hope everybody of you are safe and doing fine in this tough situation. We are all going through a new phase of life, which is the the post pandemic time. I would say we are coming out of the pandemic by the grace of the God, and I wish you all safe health and. Along with me, there's a slight change in the title today. Along with me are my other colleagues, Dharmesh Patel and Vishal Acharya. So they will also be taking part in some of the topics, and they will be sharing their expertise in the areas of ELISA-based PK studies. So moving forward, I hope my screen is visible. Moving forward to my talk, this is the title of the talk as indicated in the brochure, which is Advancing Biosimilars from Developmental Stage to Clinical Evaluation. And we are trying to focus on few key technological advancements and some regulatory insights. And I would also like to thank my earlier speakers, especially Dr. Ravi Shankar, where he has covered an extent to a large extent on the analytical aspects. So I'll try to skip most of them, but I'll be happy to take any questions at the end of the session. So here is a brief outline of the talk we would like to focus today. So initially we will get started with the overview of biosimilars, and then a little more moving from the development side of biosimilars from the bench to the bedside. When I say bedside, it's more on the clinical spectrum. And then the key technological advancements, what we are bringing in from Somru and Ingenuity Biosciences. Then certain topics of project management and CRO requirements for outsourcing I have not listed on the slide, so hence you see them in red. So I'm going to touch upon a few areas which are very critical for any product development, especially biosimilar development, how project management plays an important role. And then a lot of companies do look for CRO partners. So what are the CRO requirements for outsourcing that I would be broadly touching upon. They will not be represented on the screen, but I will be giving you a little overview about project management and CRO. And then moving forward, there will be slides on ELISA-based studies focused on, especially on the pharmacokinetic studies using ELISA platform. How we do method optimization, how we do method validation will be covered by my colleagues Dharmesh and Vishal. Dharmesh will especially focus on the PK assay. And then the case study specifically taking PEG GCSF, since it is more of a academic talk, we wanted to keep more focused on a particular case study. So we'll give you some insights into how a PK assay, a pharmacokinetic assay, which is more for clinical studies, as how it will be developed in a clinical CRO setup. And then followed by, once the assay is developed, how the study samples are going to be analyzed. That will also be covered by Dharmesh. And then we will give you an overview of immunogenicity assays, especially the ligand binding assays, and the cell-based assays which are done in the laboratory. And then the strategy for immunogenicity testing, Similarly, the method development of screening and neutralizing antibodies, and what are the method validation requirements for immunogenicity. So broadly, the topics will start from biosimilars development, starting with the overview, development, and then touching upon some advancements, and then specific areas of work like the PK, ELISA-based PK, then how practically it is done in the laboratory, and then immunogenicity studies. I hope the flow is acceptable to all. And I see if there is a chat session, you may also leave your comments so that I get a chance to see if I have to stop in between. Dr. Somasundram, just that I'm not able to see. I'm able to see only the screen. I'm not able to see you. Earlier, I was able to see you also. I mean, the participants. So, is it okay to continue like this?
Yeah, it's okay because I can see you and I can see other two as well.
Okay, great. Sure. Yeah. Yeah. So here is a biosimilars overview, especially if you look at biologics. So these are essentially very effective, which are life-altering therapies. And if you see the regular biologics, these are highly expensive as compared to generics. When I say expensive, and there is a limited access globally because of the expensive nature of these biologics, especially in the healthcare industry. And then the many so the bright side of it is a lot of these originators or innovators, they are nearing their end of the patent life. That means there's a scope for these molecules to be developed again, and then that's where is the biosimilars coming up. So certainly there is a high demand for the high quality biological therapies in terms of increasing the affordability, in terms of increasing the accessibility. So especially the I would say the third world countries or the second world countries or the BRICS nations, where the affordability is a major concern because the insurance is not well established in countries like for India especially, and where affordability becomes very challenging. So having an alternate version of a highly valid biological therapy like a biosimilar molecule would be a big blessing in terms of satisfying their specific needs in the health industry. So I'm sure this terminology you have gone across many times, but I will quickly walk you through what exactly is a biosimilar. So a similar biological or a biosimilar medicine is a biological medicine that is similar to another biological medicine that has already been authorized for use. It does not have any meaningful differences from the reference medicine in terms of quality, safety, or efficacy. So I have borrowed these two sections. Of course, it was a bit of desktop research I have done, and I have borrowed these slides basically to tell you what exactly is a biosimilar meaning. That is, which does not have any meaningful differences. So when I say meaningful differences, as we have seen in the earlier speaking Dr. Ravi Shankar about the biological analytical similarities. So they make a significant portion of biosimilar pipeline development, I would say. And if the differences are not very analytical differences, they are still acceptable. So that's what I mean to say, that they do not have any meaningful differences. For example, you have seen in the slide glycan differences. If there are any significant glycan differences, there are also post-translational modifications. If these differences are subtle and minor, then they would not impact the final clinical efficacy of these molecules. So in that way, they would be acceptable. In case if there are significant differences at that level, it could be at the clone level, it could be at the purification level, it could be at the storage level, stability level. So all these would eventually impact the efficacy of the molecule. If one is able to demonstrate similarity at a very early step, right from the early from the purified stage onwards, then it is a highly acceptable, and the data can be cleared by the regulatory authorities. Now, in terms of some of these terminologies, it's better to get a hang on these terminologies because we actually use these words routinely in our industry, in the biopharma industry. So like a biologic product is something which is, it could be a virus, it could be a therapeutic serum, a toxin, antitoxin, vaccine, blood component, etc. So all these come under biologic product. Now, if you look at the word originator or innovator, this is sometimes it's also called as comparator. It is also known as a reference molecule. And then this is a novel biological medicine that has been patented. So as I mentioned to you in the earlier one, a lot of molecules are going off patent. So this is what is a key thing that needs to be borne in mind when we are studying any particular molecule of interest. If any company has a vision to develop a biosimilar, they would initially look at the molecules which are going off patent or which have gone off patent. So we say that those are innovator molecules or reference molecules. And of course, the reference molecules, it comes whether it is a US reference molecule or a European reference molecule, depending upon which regulatory agency has given the approval for that molecule. So geographically, as well as the regulatory agency approving it, so the name would be given to it. So that is the US reference or the EU reference. And similarly, the name innovator or comparator is one and the same. And in terms of the biosimilar, so there are other synonyms for it, which is like follow-on biologic, which is most widely used in the US. And then in Mexico, it is called biocomparable. And in Canada, it is subsequently entry biologic, which is used in Canada. So which is also bioequivalent biologic. So there when I say bioequivalent biologic, it is as I mentioned to you in the earlier slide, there are no significant meaningful differences. So hence it would qualify as a biosimilar. So if you look onto the right side in terms of a little elaborate definition of the biosimilar, which is a biologic medicine with identical primary amino acid sequences to an originator medicine. So when you see at the primary amino acid sequence, for example, when you do either an N-terminal sequence or whether you do a peptide mapping experiments, you actually get deep into the amino acid information of these biosimilars. Most of these, I mean, all the biosimilars or monoclonal antibodies that we're going to be talking are primarily the monoclonal antibodies having molecular weight of roughly about 150 kilodaltons. And when you look at the sequence of this, right, the sequence has to be obtained. One is from the clone level, we will get the sequence, and that is the genetic sequence. But if you would like to get the entire amino acid sequence, we do high-end analytical characterization services like intact mass spectrometry, which will tell us what is the molecular mass of the molecule. And then if you are looking at modifications, one has to do without stripping the glycans. But if you're looking at the amino acid sequences, we need to do something called as the peptide mapping experiments, which involves digesting the protein with enzymes, with different combinations of enzymes, and then doing high complex experiments on the mass spec. There's another way of getting the amino acid sequences by N-terminal sequencing, where if one is interested in looking at the first N-terminal 15 amino acids, one can also do that. So essentially in biosimilars, the first level that starts is having the parent or the innovator sequence and the molecule that is under consideration for development should ideally should have identical sequence, amino acid sequences between the innovator and the reference and the biosimilar molecule. Now in terms of the sequence identity, one is, as I mentioned to you, high-end mass spec, but there's also NMR-based techniques which are getting more popular now. If one wants to look at the structural conformation of these molecules in terms of the similarity maps, it is also by NMR one can get the fingerprinting information of these biosimilars. And in terms of biobetters, these are called as the second generation biologics. They're also called as bio superiors. And then similarly, there's another terminology which is called copy biologics, which is an alternate biologic or biopharmaceutical which is not subject to regulatory approvals. So these are some of these terminologies what we will be seeing in the industry very widely. And here is a snapshot of two windows. What you see on the left side, you see simple biologics like EPO, GCSF, then IF and alpha, IF and beta. Then on the complex side, you see all monoclonal antibodies, which I was talking about in my earlier slide. That is, these are complex biologics, which are more on the biosimilar side I'm talking about now. And we will go a little deep into these monoclonal antibodies, what is the sequence, etc. But what you see on the complex biologics is the commercial names or the proprietary names as per the manufacturer. And these molecules, some of them are also being manufactured or in the development pipeline in India. And we at Ingenuity Biosciences, we do have methods for performing some bioassays, clinical studies, especially in the domain of clinical spectrum. We do have validated methods for a majority of these complex biologics. And in terms of people who have moved from small molecule industry into large molecules or from generics, because this is what is most of the CROs have been doing, moving from small molecules to large molecules. So the small molecule is the generics, the large molecules, especially in case of in the CRO industry where we are engaged in services, we call it as biosimilar services. And in the small molecule, proof of quality and bioequivalence. There is no substantial clinical data that is required. So essentially, it lies on the bioequivalence and the proof of quality. And then there's the reference to originators data is also required. Whereas in terms of the biological molecules or the biosimilars, right? So there is a different manufacturing processes that can result in different yield differences in the end product. So when I say manufacturing processes, for example, fed batch and perfusion, there are two different ways of manufacturing the biosimilar compounds, and there is certainly a difference or differences in terms of their attributes, in terms of the total yield. So it has to be critically monitored and evaluated at every single step of the development. Then in the biological medicine, after the quality of a biological medicine is demonstrated, some non-clinical and clinical studies are necessary, depending upon the regulatory agency to which we are submitting. For example, when I say non-clinical, it is a preclinical laboratory work I'm focusing on, and also in terms of the clinical studies, from phase 1 to phase 4 studies have to be meticulously carried down, so that we once the laboratory experiments, R&D work is done, initial test batches are completed, when the clinical material is obtained, they need to be evaluated, and then studies have to be conducted using on the clinical study material. And then the immunogenic response cannot be predicted, so therefore it must be tested. But however, there have been some software simulation programs have been coming up, but most widely which is accepted is the ones which have gone through the rigorous drill of preclinical and clinical studies. So in terms of the molecules which are going off patent, once again this slide is borrowed. I put the reference at the bottom. So here you can see the molecules which have gone off patent from different timelines, from 2020, 2010 to 2015, 2016 to 2020, and then post 2020. So especially this is more interest from an academic perspective. Students who are passionate of getting into biopharma industry, there is a huge potential which is already being harnessed by CROs currently, and also product-based companies are heavily focusing on biosimilar development. If you see major pharma companies in India, and one of their major pipeline or the fast developing pipeline is biosimilars, I would say, because the risk of failure in biosimilars is significantly lower as compared to a drug development industry, where the risk of failure is going to be a very expensive task, whereas in biosimilars, it's a low-hanging fruit. It is definitely complex. It is not that it's a very straightforward path, but as compared to drug discovery, there is a reference which is available. So there's a guide or there's a map which is available for the scientists to start working and developing their assays, validating their assays, and then submitting the regulatory packages. So it is far more successful venture to get into biosimilars. So I feel the students who are going to be getting enrolled into this program of biosimilars, it's a visionary thought of the University of Nipur, especially Dr. Ravi to bring in this kind of a blend between industry and academia, and giving us an opportunity to share our experiences with the students who are interested in taking this course. Then moving a little forward on this, what should we compare with the reference product? Like, for example, in the in vitro studies, which is generally performed to assess any possible differences in the bioactivity, right? So in the in vitro studies, what we do is, binding to the target antigen, binding to the receptors, and then if it is a Fab associated functions, especially the neutralization, the receptor activated or receptor blockade, then the FC associated functions. So all these have to be assessed in the in vitro studies, which most of the companies, most of the biosimilar companies, either they have a R&D division where they focus on this and it is done, or if these were to be outsourced. Now comes a little bit of the project management issue here, or the importance, not the issue, I would say, because as I mentioned to you, the biosimilar development, many of the companies are in a big race of launching the biosimilars, and especially early to launch gets a significant benefit. First to market gets a significant benefit when launching these compounds, when launching these biological molecules. So having developing these assays takes some significant amount of time. If there is a partnering lab or a CRO lab which has established these assays, for example, we are looking at Fab associated functions, the neutralization assay, for example. If the neutralization assay has to be developed, so the timeline on a tentative scale would be somewhere about 6 months in a product-based company, I would say, because they have multiple products working on. And right from procurement of the cell lines, right from procurement of kits, optimizing those experiments in the laboratory. So this is a significant amount of time that gets invested. Whereas when these are being outsourced, so a lot of CROs keep their methods ready. That is, the method development time and the method validation time is significantly cut down, so that this can be passed on to the client. The timeline advantage will be passed on to the client. Of course, it does come with a certain associated cost, but it is eventually a big victory for the companies who are participating right from the early stage of that development, so that the initial development can be transferred to the CRO where we develop it. So, for example, the way we do it at Ingenuity Biosciences, I would say, it is depending upon we initially start with something called as a feasibility studies. Let us say that any biosimilar company initially approaches asking for a specific assay or a specific molecule, the expertise of the scientists in the team, then we would actually run through a little matrix. So we have a grid, a matrix grid. We have a different scoring algorithm, or which is manually given. So we assign number, a metric to each of these attributes. For example, when the feasibility comes in, we initially look at availability of the kits. We look at the availability of consumables, availability of a valid method in the lab. What about our scientists having hands-on experience in that? What are the challenges in that? For example, omalizumab. It is as compared to a PEG-GCSF. PEG-GCSF, the linearity is quite straightforward to establish in the PK study. Similarly, in the immunogenicity study, if you look at omalizumab, it comes in 2 doses. So it takes a little amount of additional work to set the hands on the omalizumab. But when we are running through a feasibility grid, so we also assign a score saying that this is a critical molecule. So we give a higher unit of scoring. So eventually, what comes out of this the scoring matrix is on the feasibility test. When we revert to any client saying that there is a high level of feasibility of we doing this, or capability, it is a reflection of capability of the CRO in simple terms. So we say that yes, the feasibility score has come high. We can handle the study quite efficiently right from the development stage till the clinical stage. So when I say clinical stage, the phase 1 studies, we can completely handle it with the resources what we have built in our lab, and also the resources what we are getting from our partner lab overseas. So that is in terms of the in vitro studies. And in terms of the safety pharmacology, reproduction, mutagenicity, this is more on the preclinical side, so these are generally not required. And coming to the in vivo studies, so these are dependent upon the need for additional information, that is the residual uncertainty and the availability of a relevant animal model. So these are also on need basis. So I'll move to the next slide. So this is a quick overview of what you see on the right side. Is a little cartoon of a monoclonal antibody having the light chain, heavy chain, the constant region and the variable region. What you see on the top, the white projections on the right side is the antigen binding site having the CDR domain. So the CDR domain is the one which plays an essential role in binding to the the target site. So the epitope, that is whatever is present on the monoclonal antibody, plays a significant role in having a complementary binding to its target. Now, when we look at the nomenclature of these monoclonal antibodies, it takes a little bit of effort for beginners to understand these names, especially if you look at the examples like adalimumab, pantalizumab, denosumab, rituximab, infliximab, cetuximab, etc. So this is, basically it has been taught in graduation or masters, the origin of these names. For example, if you see, if it is, if you look at the word U, the adalimumab or in the pantalizumab and the denosumab, the alphabet which comes before mab is U. So when you see that prominently being there in the names of the molecules, so you would say U represents that it is a human monoclonal antibody. That means it's entirely derived from human source, which is very critical. That is, it would have minimal immunogenic response as against the one which is developed from a mouse. Like for example, in the row 4, you see an entirely murine source which is having MO in its nomenclature fitting, that is pintumomab or vectumomab. So where it has come from the murine source. If it is a chimeric, which is a fusion between, it's a variable regions coming from murine and constant regions coming from human. So like for example, the rituximab, infliximab, cetuximab, etc. So these are all chimeras because they're coming from 2 different species. And if it is completely humanized, that is, it is mostly from human except for the binding region. So the trastuzumab, bevacizumab. So the name, components U, XI, or ZU, these are very important. It gives an indication about what is the source of these monoclonal antibody development, and also the initial immunogenicity one needs to plan for these molecules. Here is a quick snapshot of the names of these molecules once again. Who is the innovator of these molecules? When was their initial approval, and when is the revised approval of these molecules? So if you look at rituximab, it was initially approved in 1997, and there's a revised approval happening in June 21. Similarly, PEG-GCSF 2002 in April 19. So this is a mistake there. It shouldn't be that way. Okay. And then cetuximab, adalimumab, so on and so forth. You see the brand name and you see the innovator companies. Coming to the little overview of the biosimilar development, what the way it begins is first is getting the RFP or the reference molecule, sourcing it, and logistics. But prior to this, prior to the RFP sourcing, I would say there is a lot of effort that goes in the laboratory, the development laboratory, the R&D lab, of having the clone development and clone evaluation. This takes a significant amount of time. And then then the next step is validating the amino acid sequence, the amino acid verification. Did it translate in the right way? Are we getting the right sequence of amino acid without any alterations, without any mutations into it? And then establishing the quality target product profile, which is the most important aspect, which helps in setting up the specifications. The quality attributes are another significant thing, which are sort of guidances or roadmap for the product development in a product-based company. Once these are established, then that means the specifications are set. People know what to look in their molecules, what sort of flexibility or what sort of offer has to be made during the analytical assessments. So similarly, the methods will be developed. When I say methods, it's all the analytical methods. There's a battery of about 12 to 15 analytical tests which need to be performed. And so in my past experience, I was heading a national laboratory. This was a BIRAC funded lab where we had extensive end-to-end high-end characterization services offering to clinical labs or even to development labs. So when you look at these methods, there are two kinds of methods. One is a release method, one is a characterization method. Most of the release methods are heavily scrutinized by regulatory agencies because since we are, as I mentioned to you earlier, the biosimilar molecule, it has to be a replica of the innovator in terms of the analytical aspects. So, but however, there is a certain degree of flexibility that is given, like in terms of the glycan variations. There are certain modifications which are very stringent, not much of room for any alterations there. But however, there are certain modifications where there's a slight window when we, especially when we are doing the quantification. There is a window that is set. If it is falling, I'm sorry, if it is falling within the window, then it gets accepted. So those are all heavily monitored, which are release assays. And the release assays are very critical because batch to batch, when batches are being manufactured, every batch release has to strictly go through or adhere to those guidelines, to those specifications which are listed during the method development and method qualification process. And once the method gets developed and qualified, it also gets transferred to the QC laboratory, where every batch has to be very rigorously tested for the release assay methods. On the other hand, there is also a characterization method which is not as stringent in terms in the eyes of the regulators, because we don't have to go through the extensive battery of method qualification in that. When we talk about methods like analytical ultracentrifuge, when we talk about methods like SECMOLs or high-end mass spectrometry, disulfide bridges, in these cases, it's mostly these are our characterization assays. And during these characterization assays, the concept of method qualification or method validation is not applicable to in these cases. So just to summarize in the originator, so it starts with the cell line development, which itself is a intense, laborious work, high intellectual extent of thinking will be going on into this. And then, of course, the media optimization. The media optimization is essentially for getting the right amount of yield, and there are different high throughput techniques which have come up now in order to screen the media, in order to screen the clones as well. There's one such technology called AMPR, where one can screen the clones at a high throughput method. The conventional method is to screen the clones and then do the analytical characterization, especially on the glycans. So they cleave the glycans and look for the glycan profile initially. That is something which is routinely done in any development laboratory. And then once the initial clone is established, once the media optimizations is all done, and there's a good amount of yield that is coming from this batch, then it goes to the next level, which is the downstream development, where the purification happens. And then again here, analytics play an important role. Post-cell protein, post-cell DNA are the process-related impurities that need to be critically evaluated. So once the product development passes this step, it goes to the engineering runs, and the GMP production batches happen. It starts from 50 liters or 10 liters also. We have done with 10 liters, we've done 50 liters material which were used for preclinical studies. And then it goes through a series of preclinical tests. As I mentioned to you earlier, the toxicology studies go on, and also some of the preclinical studies which are involving the PK and immunogenicity studies will be performed, and then followed by the clinical studies. Once the molecule, the originator, goes through successfully through all these steps, it gets submitted to the regulatory authorities. The data gets evaluated critically, and then it passes the stage of reaching the approval. So this is the entire journey of an originator. Whereas if you look at from a biosimilar perspective, the later half of it, most of it essentially follows the same. But there's a major exemption that one gets from preclinical step here, and then on the clinical side to a certain extent, but the majority is heavily banking on the analytical characterization of it. If there's analytical characterization is found to be similar, one can shrink the time of the preclinical and also the clinical phases of the development of the biosimilar. So keeping the audience in mind, students who are interested in pursuing their career in biosimilar, there are if you are a molecular biology background person, you're very passionate of doing a molecular biology based work, then I would suggest cell line based laboratories are the key focus areas you want to keep your attention on. Similarly, if you are more of an analytical mindset, you are more savvy of new technologies coming up, you want to set your hands on different analytical platforms, then definitely it is the analytical division in the development pipeline of biosimilars or biologics. And similarly, in the the same molecular biology person can also fit into the upstreams, where the same operations happen. Apart from clone development, it is also clone evaluation, clone screening, and then in the downstream, it is mostly on the purification process. So if you have interested in the purification process, I would suggest to look for opportunities in the downstream in the biosimilar industry. And if your interest is overall looking at the timelines of delivery and moving from cell line development to approval, coordination across different teams, coordinating with the with different vendors, coordinating with internal teams, different stakeholders, then project management is one of those key areas which sorts of unites different stakeholders and keeps them on the time track. So that's the project management team. The project management team also coordinates with the CRO industry to see which specific program has to be outsourced, considering so that it fits into their specific timelines. Yeah, and there's been a recent advertisement, or I would say a recent announcement, not advertisement, a recent announcement from our side, from Vida side, where we have been working with one of the preclinical labs in India. So our partners, CX partners, have invested into BioNeeds. So BioNeeds is another preclinical CRO based in Bangalore. So you can see the list of services in terms of from the drug discovery services to the phase 4 studies. So they all come under now Ingenuity. So we offer mostly the in vitro based assays, the cell based assays or immunogenicity assays, the PK/PD studies in the preclinical domain. Similarly, on the host cell proteins, if a biosimilar compound it is going for regulatory submissions, the regulators do ask for specific antibodies to be developed using or antibodies to be developed using their specific compound. So taking the test compound or the test material, so it is induced into the animal and then the antibodies get generated. Those antibodies are purified, where it can be used for defending when we are submitting it for regulatory authorities, especially on the immunogenicity testing side. Similarly, on the host cell proteins, host cell DNA, the same thing is applicable. When we are, there are also commercially available kits for host cell proteins which are commercial, which are more generic. So the antibodies that are present in those are not only product specific; those are more generic. But whereas any company which is keep investing its efforts in the development of their own host cell protein kit. So essentially, what they do is they take the compound and they do a cascade immunization process, either in the mouse or in the rabbit, collect the sera, do immunoaffinity purification, and then quantify the extent of the host cell protein that is present post purification of the material. Similarly, this is another repeat again, like in terms of cell banking, cell line characterization, what are the areas that we will be offering services. Similarly, in the preclinical domain, like the bioidentity assay, the biopotency assay, toxicokinetic assays. Currently, the polyclonal antibody generation in mice, it is global. So, but these are high-end and niche services, which these are generally a long-termish activities. But certainly these are to the marquee clients. These are offered. And focusing a little more on the technology advancements, at our place we have 3 major innovative technologies. We call this as MapY platform, Igris platform, and Intelity. So when I say these are innovative technologies, these are more futuristic technologies, I would say. When I say futuristic technologies, for example, in the area of upstream and downstream, in the upstream, perfusion has gained a significant momentum as against fed batch reactors. Similarly, in the downstream, QBD based quality by design, which is the QBD, that's based downstream, has gained significant momentum. But coming to the analytical space, in the functional characterization assays, especially the ELISA based, PK based immunogenicity assays, there are proprietary softwares, but there's a high, it's another high demand complex field. So getting into advanced technologies like using proprietary customized softwares plays an important role and provides a significant fast tracking of the molecules. So I'll walk you through what these platforms are. Essentially, the MapY, Agares, and IntelliB. And overall, in terms of the experience, we have over 120 validated methods for different biosimilars, which have been submitted in global arena in the US FDA and the EMA, and also participating in the strategy meetings, especially the type 2 meetings of the US FDA. So our team has experience. When I say our team, it is our counterpart Somru team with whom we both work together. So we have collective experience in helping our clients with their regulatory submissions. So overall, it's the end-to-end, right from R&D to preclinical and phase 1 clinical trials. That's what is the entire bread and butter of our organization, from the R&D to the phase 1. And then, of course, VIDA, I'm sure a lot of you have heard about VIDA Clinical Research. They offer services in the clinical domain, and they have been pioneers in the field of small molecules. But now with the Ingenuity coming up in the area of large molecules, we are hoping to have a very good journey, and we'll be glad to get connected to any product-based company who are representing in this conference. So in terms of these three that I mentioned, they're essentially used for biocompatibility testing, and also in terms of the bioassays, they are deployed. And here is a snapshot of what you see, the methods that got developed in our counterpart laboratory, starting with the product name on your left column. And then what you see is the list of all the maps on the top: cetuximab, rituximab, bevacizumab, so on and so forth. And then as you move from top to bottom, starting with the detection method, it is a calorimetric, it's a ELISA-based method. There are also other methods like mesoscale discovery, which is also more widely used in the industry. We also plan to get the chemiluminescence method soon, but as of now, you can see that's the current method that is available. And in terms of the precision, what is the percentage CV the scientists have achieved in the laboratory? What is the accuracy of these kits? The sensitivity, if you look at, for example, rituximab, the sensitivity is as low as 55 nanogram per mil. And if you see the hook effect, so I don't want to walk you through entirely through all these maps, but in a nutshell, to say that this slide reflects the capabilities in terms of the assay development, in terms of assay validation that the team has achieved in making this ready to be used by any biosimilar company who are envisaging to develop their methods with us. So MapY technology, it is a next-gen antibody discovery solution. So if companies who are in the early stage of discovery, so they would invest into a technology platform called MapY. Here the key advantages of this will be, there'll be hundreds of high-quality leads against the diverse epitope. So for example, if you look at, as I mentioned to you earlier, in the drug discovery industry, like the innovator that I've shown you on the top, the significant amount of time goes in clone characterization. But in terms of, when they are developing this molecule, but developing against which specific binding epitope, that plays an important role. So here it goes through several iterations on the software to produce high quality leads. So that is the unique advantage of this. And in terms of the faster discovery, that is from immunization to purified antibodies, it will be in as little as 3 months. So that's a significant advantage of it. And the entire database, what is present here, it is mined using untapped antibody in the NGS database using machine learning algorithm. So a lot of time and effort has been invested in developing this MapY technology. And the rational antibody design, so it utilizes residue to residue interaction database to design antibodies and explore the affinity maturation of fully human antibodies. So it has been tested against several clones so that the high quality lead against the epitope is successfully established. And coming to Agris, this is a lab informatics and automation solution platform. It's an integrated bioanalytical method validation software which is readily used in the PK development, in the ADA development, in the biomarker, and other biosimilar assays. So Agris software, it enables the scientists to perform method validation and also statistical analysis in a regulatory compliant manner. So it's a one-stop solution. Let's say that with the plate reader, once the scientist completes acquiring data on the plate reader, he would pass on the data onto the Agris software, where it would do the complete statistical analysis and provide a report which can be used for regulatory submissions or in a regulatory compliant manner, I would say. And also the software is being developed as a web-based application, so that means it can be hosted on the web. And the front end of the software provides a rich user interface. So there are a lot of fancy tools that are embedded into the software. And yeah, in terms of, there's also a built-in open interface module which allows seamless importing of data from other third-party softwares or links. So that's an advantage of Agris. It can be seamlessly be used for cross-platform integration. And moving forward, there's also a lab automation robot. Right? Currently, what one does, generally the typical life of an analyst in the lab begins with pipetting, doing initial sample processing, preparation of standards, the HQC standards, preparation of different buffers, and then handling samples from the refrigerator, and then reading it on doing the initial reactions, binding, washing, and then doing the secondary antibody incubation and followed by detection. So a lot of sample prep steps are involved before it goes on to the plate reader. So there's also another version which is the automation robot, which essentially does from the sample handling step till the sample readout step. So that's also gets integrated with the Agris software. And the most important thing is the fingerprinting solution, which is called as the IntelliB, which is the next gen solution. So this platform, the IntelliB, is based on more than 2,000 antibodies which are generated against various innovative drugs. So these are curated ones where the platform is actually data has been generated using 2,000 odd antibodies here. These antibodies bind to innovator and biosimilar products in an ELISA based manner, so to give a meaningful fingerprint like analysis. So if you see here the cartoon on the right side, these are the antibodies what we are talking about, which are binding to the specific regions. So when you look at your innovator and your monoclonal antibody of interest, that's your test product against the innovator. So essentially, the IntelliB would give a fingerprinting like solution when it is run through this particular piece of software, or since it is the experiments have been done in the lab, the antibodies have been generated, have been run against these innovators. So getting a meaningful fingerprint like analysis is what is possible with IntelliB. I will pause here now, and I will pass on the next slides where my colleague Dharmesh will be talking, and then followed by Vishal. So I'll be happy to take any questions at the end of the session. Dr. Somsundram, please let me know if you want me to take any questions now.
I think we can have it by the end of the session.
Yeah. So over to Dharmesh.
Okay, good afternoon all. I'm Dharmesh Patel, working as a senior principal scientist at Ingenuity Biosciences Private Limited. And thank you, Dr. Ravi, for handing me over this session for further discussion. So I'm going to talk about the ELISA assays for the PK studies, or PK ELISA assays, which is required. So let me make it clear what are ELISA PK assays. So as we know, the ELISA is enzyme-linked immunosorbent assay technique, which is used for detection of either antigen or antibody. Okay. So when we say PK assay, so this is with respect to the concentration of biosimilar or reference product in the blood when it is administered into human during clinical studies like clinical trials, phase 1, phase 2, phase 3 clinical trials. The drug is administered, okay, biosimilar drug, large molecule drug, or during biosimilar study, like to prove the bioavailability equivalence of biosimilar with respect to reference product. So the samples which are collected, blood samples, human serum samples, plasma samples. So to estimate the concentration of that product, biosimilar product, inside the blood, so that those assays are called as PK assays. So when we say enzyme-linked immunosorbent assay, there are 4 different types of ELISAs, like direct ELISA, indirect ELISA, sandwich ELISA, and the competitive ELISA. So out of that, I am mostly because mostly the sandwich ELISA is being used for the biosimilar estimation. So let us have a view on sandwich ELISA. Like what happens, the coating of the plate happens with antibody, which is immobilized antibody. And once that is happened, the excess of the solution is removed. Then whatever surface that polystyrene plates are used for the coating of the plate. Now plates are being coated either with antigen or antibody. This is totally depends on the type of analyte. If type of analyte is like antibody, then antigen is coated. If type of analyte is like antigen, then the antibody is coated on the plate. Once to block the unutilized surface, the blocking is done, like Bowen serum albumin is most of the times used to block the uncoated surface. Afterwards, the sample which is containing analyte, which may be either antigen or antibody, is added to the plate and is kept for the incubating temperature, maybe at room temperature 20 to 25 degrees Celsius. Then afterwards, it is again excess solution is removed and washed, and then antibody which is specific to the antigen, which is linked to the immobilized coating agent, is added. So these antibodies are generally secondary antibodies or antibodies which are linked with enzymes, and it is again specific to antigen. So it will attach with antigen. Then again excess solution is removed and washing is done. Then enzyme substrate is added, which forms the color, and the color absorbance is measured, and it is dependent on the concentration of analyte. So that is how sandwich ELISA works. The next slide. So after discussing about the principle of ELISA, so how do we start the method development and optimization at CRO? Like uh, uh, at Ingenuity, we generally use the kits which are developed by Biosciences. So the ELISA reagents and kits and antibodies are developed by and those kits, validated kits, are transferred to Ingenuity Biosciences. And there, the optimization of method occurs with respect to the study. When we say with respect to the study, with respect to the dose of the biosimilar in the study, what are the anticipated concentrations, and based on that we do optimization of these kits with respect to our need of assay. So we have to search for the biosimilar literature with respect to the reference. For example, pegfilgrastim is a biosimilar product, So for that, we will go and search into Neulasta, which is the innovator product of filgrastim. For filgrastim, the innovator product is Neupogen. So we'll go and search and we'll try to find out what are the low levels of drug after administration. And based on that, we have to find out what is the Cmax of the drug. And based on the Cmax, other decisions are taken for setting up the linearity range. So please, next slide, please. So once the ELISA kits are received, they are stored at appropriate temperature because some reagents are to be stored at 2 to 8 degrees Celsius. Some reagents may require storage at minus 20 degrees Celsius. So such storage facility we should have. Then to create the calibration curve and quality control samples, we have to use both kind of formulations like biosimilar formulation as well as reference formulation, and we prepare stock solution from both the formulation. Now, when I say what are calibration standards, so calibration standards are 6 to 7 standards. So it will give us those response relationship, and based on the calibration curve which is constructed, the concentration of unknown sample is being calculated. So quality control samples are again a known concentration samples, but they are not the part of calibration curve, and they are used to check the performance of the calibration curve. So based on the predefined acceptance criteria of quality control samples, the curve is either accepted or rejected, and based on that, the further analysis is accepted or rejected. So we use the stock solutions prepared from both reference formulation as well as biosimilar formulation for preparation of quality control samples, while for preparation of calibration standards, reference formulation is used. The calibration curve standards, calibration curve is prepared by having 8 to 10 calibration standards with different concentration range. Then we also prepare anchor calibrators. Now these anchor calibrators generally prepared nearby upper limit of quantification and lower limit of quantification range, so that they will support the non-linear curves, because many times our equations which are the calibration curves which are formed in ELISA, it's non-linear curve, and non-linear curve to support those curves, some supporting standards are added. So those are known as anchor calibrators. So how to set the ULFQ, that is upper limit of quantification calibration curve. So it is approximately 2 times of Cmax of the formulation which we are going to use in clinical study. While the lower limit of quantification, the requirement which is specified by the regulatory is at least 5% of the Cmax or lower than that. So that is how range is decided. For example, a range Cmax is 100 nanogram per ml, then our expected ULOP should be around 200 nanogram per ml, and the lower limit of quantification, the expected is either 5 nanogram per ml or less than that, and rest of the standards are adjusted between these two standards, and thereby total 8 to 10 standards are prepared. So we are, as we have discussed, there are two sets of quality control samples. One is prepared from reference formulation, another is prepared from biosimilar formulation. Now the quality control samples are of, we have to prepare at different 5 levels. During validation, we need total 5 level quality control samples. That is, one is ULOQ-QC, which is having concentration equal to ULOQ. Then high QC, HQC, which is having concentration 75% of ULOQ. Then middle QC, that is MQC, it should be having concentration between 30 to 50% of calibration curve. And LQC, that is lower QC, 3 times, it should be having 3 times of LLOQ concentration. And fifth quality control sample, which is LLOQ QC, should be having concentration equal to LLOQ. So the CCs and QCs are prepared by spiking the spiking solutions in blank serum, human serum, and plasma, and that is how the calibrators and quality control samples are prepared. And it is advisable that These calibrators and quality control samples has to be frozen at least 10 to 12 hours before we do analysis, because the study samples are also comes in a frozen condition. So that has to be in a frozen condition. So that is a recommendation by regulators. Next slide. So once this, yeah. So once the method optimization shows that the ELISA cut what we are going to use is giving some satisfactory results, then we have we do prepare or follow perform some experiments inside the method development itself, and like accuracy, precision, selectivity, and some sort of stability. And once that is over, then the method optimization is completed, and we proceed ahead for method validation. For going to method validation, we have to prepare method validation protocol, as well as the method SOP is prepared. And inside the method validation, the experiments which are performed are selectivity, accuracy and precision, then ruggedness, sensitivity, specificity, dilution integrity, concomitant medication, stability. So selectivity is performed in 10 normal lots, plus in addition to that one lot each of hemolytic and lipemic serum also taken. From each lot, the prepared samples are a blank sample, lower limit of quantification QC sample, and high QC samples. And those are prepared, processed, and analyzed along with the calibration curve samples. Accuracy and precision batches are total numbers, 6 batches has to be performed. Ruggedness, one batch for one analyst as a part of different analysts. Then sensitivity is the LLFQ samples. So we have LLFQ samples in precision and accuracy batches. So we take those samples for considering the sensitivity. Specificity is performed for the structurally similar compounds. So like in case of insulin aspart, maybe the insulin, human insulin itself is a structurally related compounds, but sometimes some other analogs of insulin. So we have to check the specificity of those if it is anticipated that the study will be containing both drug as a concomitant along with the study drug. So those concomitant drugs which are structurally similar to our biosimilars, for those we have to perform whether they are interfering our analyte of interest or not. So for that specificity is to be there. Dilution integrity is again called as hook effect, where we are doing several dilution of the very high prepared QCs, which is out of the range, and we see whether after applying the dilution factor, we are getting the actual concentration or not. And inside the stability, like there are two types of stability: solution stability and matrix stability. Like solution stability is again of short term and long term stability. Short term is of bench is at ambient temperature. Long term stability is done inside the refrigerator storage or maybe inside the deep freezer storage based on the storage condition of the stock solution, and that is to be done for maybe 10 days, 20 days based on our anticipated use of the solutions. The matrix stability is nothing but the stability of analyte inside the spiked serum. So we are using high QC and low QCs, and like benchtop stability, freeze-thaw stability, and long-term stability in deep freezer is performed along with the calibration curve standard and batch qualifying samples. So once the method validation is over, so all the validation experiment results are compiled on day-to-day basis. Then and draft method validation report is prepared, and all the data generated during the validation along with method validation report, they are reviewed by quality control persons as well as quality assurance persons. And after having peer review is completed, the final method validation report is generated, which is used, and our method can be said as method is validated and is ready for analyzing the human samples. from the clinical study. Next slide. So this is one of the case study of the packed filgrastim where we have done asset transfer from Biosciences to Ingenuity. The packed filgrastim, which is having molecular weight of 39 kilodalton, is a pegylated form of filgrastim, and this molecule is used for the bone for stimulation of bone marrow to produce the and increase the WBC count. So PEG-GCSF assay, it employs the sandwich enzyme immunoassay from Sambru Biosciences using PEG-filgrastim Neulasta as innovator product. Okay, the next slide, please. Yeah. So the method is here. The anti-therapeutic protein is coated onto 96 well plate. Then the therapeutic reagent present in the sample is bound by immobilizing the antibody. After washing away any unbound substances, biotin-linked antibody is added. Following the wash to remove unbound antibody, the conjugated streptavidin reagent is added to the wells, and following the wash to remove any unbound conjugated streptavidin, a substrate solution is added to the wells to form the color, and finally whatever form color is there, it is measured into microplate leader. Then this assay is linear between 156 to 10,000 picogram per ml, and we have very good precision of less than 5.3%, with good accuracy between 95 to 101%. Next slide, please. So no hook effect was observed when the samples above ULOQ were assayed, and we got accuracy between 83 to 91%. Then by using this validated biosimilar PK ELISA method, study samples are done, and we generate bioanalytical study plan for that. And the analytical batch inside the clinical study is processed in a single plate with one set of CC standards and at least 2 sets of quality control samples, which is containing HQC, MQC, and LQC. Each samples are processed in duplicate and mean OD value of 2 replicates are used for the further calculation. Next slide. All the batches are processed as per method SOP, and the calculations are done using LIMS software. We are using Agiris software, and batches are accepted or rejected based on the predefined acceptance criteria into study plan. Now, any batches which are not which are rejected, we have to repeat all those samples. Even the batches which have been accepted from there also, we have to repeat certain individual samples based on predefined repeat criteria. But in any case, none of the sample will be repeated for pharmacokinetic reason, because that is not acceptable by any of the regulatory. Next slide. We have to perform incurred samples reanalysis as a part of to check in study validation. This is a within study validation exercise which checks the reproducibility of method. So we have to take at least 10% of the samples in case the sample size is less than or equal to 1000. If it is more than 1000, then for any sample above 1000, we are taking additional 5% for incurred samples reanalysis. From each subject which are randomized for incurred samples reanalysis, we are taking one sample from CMEX region and one sample from elimination region, and we have to process and analyze as per our regular method, and we have to check whether the repeated values are same to the original or not. There are certain criteria that it should be within plus or minus 30%, and at least 67% of the samples taken for ISR analysis have to be within the range. Next. So, and after the analysis, during study analysis also, the documentation is done on day-to-day basis, result compilation and draft study repository is prepared. Again, the same raw data are reviewed by QC and QA, and once the QA review is complete, this draft study report is sent to sponsor for review, and once sponsors observations are cleared for this study report, the report is finalized and signed up. So that is how we work in PKSA. So let me hand over this further session to my colleague Vishal Acharya for immunogenicity.
Thank you, Dharmesh. Just to add in here, Dharmesh Patel, he's a senior group leader at Ingenuity Biosciences. He has over about more than 16 years of experience. He's been working primarily on small molecules initially, but since a few years, close to 5 years, he's been working on large molecules also. And thank you, Dharmesh. Over to Vishal. Dharmesh, can you introduce Vishal, please?
Yes, yes. So let me introduce my colleague Vishal Acharya. He has been involved in large molecule analysis since last 15 years, and he is having a very good hands-on PK ELISA assay as well as immunogenicity assay. Vishal has also worked in cell-based assays and neutralizing antibody assays. And Vishal, please.
Thank you very much, Dharmesh and Dr. Ravi, for giving me this opportunity. Now I will be talking about the immunogenicity assays. These are the very important assays if we talk in terms of the regulatory recommendations. Because as you know, that large molecules are having the molecular weight more than what the small molecule has. So whenever we inject these large molecules into the patient or human body, our body has tendency to generate the antibodies against this thing. So the biosimilars or the large molecules or the therapeutic proteins which are being developed by the sponsors or the pharma companies should not be immunogenic. So immunogenicity is nothing but, I will define very simply here, that it is nothing but the capability of inducing unwanted immune response in human body when we talk in terms of the biosimilar or therapeutic proteins. So it is the regulatory expectations that the biosimilar molecule should not be immunogenic, and it should be safe, and it should have proper efficacy. Next slide, sir. So this is the strategy for testing of immunogenicity samples. So whenever we are talking about the testing of immunogenicity, there are two types of antibodies, or we can say that can be two types of antibodies that can be produced in the human body. Other one is just the screening antibodies, or we can say just the binding antibodies, which just bind with the therapeutic molecule which is being injected, and it will lower its efficacy. And another type of antibody is the neutralizing antibody, which might be no harmful to the severely harmful antibody, which totally it reduces the efficacy of the biosimilar molecule, as well as it may lead to the death of a particular patient. And the famous PRCA incident that you might recollect, or if you might have heard of it, that in case of EPO, which was the first recombinant human protein injected into the human, it created the PRCA condition, that is pure red cell aplasia condition, where the endogenous EPO, you also might be knowing that the EPO is also produced in the human kidney. So the endogenous EPO was also being tagged by these produced antibodies, and the patients were having conditions called PRCA, that is pure red cell aplasia, and the death of the patients started occurring. So I will be coming back to the strategy. So first of all, the test sample are tested in the assay, and if the samples, these are the qualitative assay. First of all, I would like to add one more thing over here, that immunogenicity testing or the antibody testing assays are qualitative assays, and they are not quantitative assays. Why? Because we do not have the exact reference standard against these antibodies available commercially, or it is also not feasible to develop these standards. Because if we want to develop these standards, we will have to inject this biosimilar molecule in the human population, and which is ethically not correct. So we produce these antibodies by injecting in the rabbit or other animals, and then we purify it and we use as the positive control in our immunogenicity assays. So these assays are either qualitative, or you can say quasi-quantitative or semi-quantitative assays, in where you determine just titer and report those titers in your report. So screening assays, if the samples are positive, negative, then you will obviously report it as negative. If the samples are positive, again you will go for confirmatory assay, where you have the another battery of testing, which I will come back to you what we will do in confirmatory assay. Here again, if the samples are negative, you will report them. Again, if these samples are positive, then it is confirmed that the antibody is present. Now you have to see that whether these antibodies are neutralizing antibodies or they are just the binding or the screening antibodies. So for neutralizing antibody assays, the guideline has recommended to do a cell-based assay, or if the most preferred one is the cell-based assay, and if it's not possible, then you can only go for non-cell-based assays. Then again here you determine the antibody. If it is coming positive, then again it is going to impact the clinical responses also. So you have to do the correlation of these produced antibodies with the clinical responses of the subjects in which sample you have found these antibodies positive. Also you have to see the PK and PD status of this patient, and you have to take the decision accordingly. Next slide. So now coming on to the method development for the screening assays. When I talk screening assays, it is screening antibody assay. So in first of all, we have to select the format and the instrumentation. You can do this just by simple sandwich ELISA or bridging ELISA, and nowadays there are also new technologies such as MSD, what Dr. Ravi talked about, that mesoscale discovery, and also there is a gyros lab technologies which are now available to detect this, which are the instruments to detect these antibodies in high end. So when we talk about ELISA, we can do it by simple sandwich ELISA. Just we coat the antibody drug or the biosimilar on the plate, then we detect our antibody which is present in the sample. And then the final detection of the secondary antibody which is used is the again the drug which is labeled either to biotin or to HRP, and for the color stimulation. Next slide, sir. These are the different parameters which we do in method development in our lab when a normal assay is developed. So here, first of all, after selection of format, we select the reagent, and again, one of the key parameter which is told by the regulatory to have is the positive control antibodies, which I told that it is not possible to have a true control. So these positive control antibodies are generated in the animals. Again, it is recommendation from the regulatory that these positive control antibodies should be against the test product which or the biosimilar product which are to be tested. So this is one of the key regulatory requirement. The other optimization parameters which we do in the lab, I will go in detail in this parameter. For this parameter, I will be in the validation part. So I will just quickly read out these parameters, that it is developmental cut point, minimum required dilution, sensitivity, drug tolerance, selectivity, specificity, stability, and target interference. Now target interference, just I would like to add over here that it is not applicable in each and every assay. But if you are testing, for example, say anti-bevacizumab antibodies in a test sample, then you will be having VEGF samples, because these are all cancer patients. So this patient sample will have VEGF, that is vascular endothelial growth factor. So this VEGF will interfere in testing of these antibodies, and it may give you false negative results. So this target interference is one of the key or important parameter to be tested in development as well as validation. Okay, now coming on to the method development of neutralizing antibody assays. So here, as I told, that cell-based assay is recommended. So what we do in cell-based assay, so I will again give example of EPO over here, that is erythropoietin. So when you are testing the sample of neutralizing antibody that is having the NAP, that is neutralizing antibody against EPO, you will stimulate these cells with the drug. So if the cells are responsive to the drug, that is erythropoietin. So in presence of drug, the cells will proliferate. And if the antibody is present in the sample, so it will get bound to the drug, and there will be a conjugation of drug and anti-drug antibody in the sample itself. So when now you will put this sample in the cells contain in the plate containing the cell will not be able to grow. Why? Because the drug is bound to the antibodies, neutralizing antibodies produced. So this is the basic principle of neutralizing antibody assays, that is the proliferation based cell based assay, and the final reading, the final endpoint result will be either a fluorescent or colorimetric based result. So if the number of cells are more, you will have the color more intense color, and if the number of cells are less or low, in case of the presence of NAT, the intensity of color will be low, and the reading will be taken in either MSD based platform or the plate reader. Next slide. Again, the same parameters what we see, what we did in the screening antibody assays will be done over here, except that here the most important will be the selection of cell line. So the cell line which is the responsive to your drug should be selected, and the reagents or the media in which this particular cell line grow are to be selected. Again, selection of positive control. And here, one of the other parameter apart from staining antibody that you have to determine is the optimization of activity curve. That is derivation of either EC70 or EC50 values. That EC70 is nothing but the effective concentration of your cells or of your curve when you have the curve in the linear range of 70%. And EC50 is nothing but the midpoint of your sigmoidal curve. Next. Okay, now coming on to the validation requirements for immunogenicity. So for screening antibody as well as neutralizing antibody assay, the validation requirements are almost more or less same, and there are little differences, but we can say that the requirements are same. So main parameter, now I am coming to the screening cut point, and I will explain you a bit in detail what is screening cut point. So here these are qualitative assays. So you have to derive a screening cut point. It is the value above which the samples will be said a positive sample, and the samples below the value, it will be termed as the negative sample. So these values are derived by running 50 drug name samples from different from the subject population, and it will be run in a balanced design having different test parameters, different rates by different analysts on different days. So once data is generated, this data will undergo a statistical analysis, and this statistical analysis facility is again available at our lab in our Agaris software. Otherwise, this statistical analysis is a bit tedious when you do it by yourself using the available softwares on net. And again, the software should be 21 part 11 compliant. So it is a bit tedious exercise. So again, this is advantage of Agaris software, that where cut point is calculated automatically. So here the recommendations I have mentioned in my slide. What is the regulatory recommendations? So the fixed cut point is a cut point which is just 1.645 into SD of the test value. But this is again discouraged by the regulatory agency, and the preferred one is the floating cut point. This will here you have to have just the negative control of your plate, and you have to multiply a correction factor or a factor which is derived from the statistical analysis, and that particular value will be a floating cut from which all the samples will be reported as positive, and below which all the samples will be reported as negative. Now coming on to the confirmatory cut point, where you have to confirm the samples are whether positive or whether it is true positive or not. So here you have to add the drug in excess amount in your samples. So if the antibodies are present, it will bound to the drug and the signal inhibition will occur. So this signal inhibition will say you that whether your sample is true positive or it is a false positive. So here again the same 50 samples we have to run with the high concentration of drug, and it will again undergo the statistical analysis, and here the false positive rate is 1%, while it was 5% in screening cut point particular derivation. So then coming on to the titer assay cut point, this cut point is almost similar to the screening assay cut point, and there is no much difference. Next slide, sir. Again, here we will have precision experiment where we will have our quality control samples prepared, and we will run inter-assay and intra-assay precision. And this will be just evaluated here by the percent CD criteria, that it should be less than or equal to 20%, and for negative percent, it should be around 25%. Because here there is no control, we will not be able to measure the percent accuracy that we were able to do in the PK assays. Here also we can do inter-instrument and inter-operator precision if it is relevant. Next slide. Drug tolerance is again an important important assay parameter. Here it is the importance of this assay. Whenever the drug is given to the patient, it will stay in the human body. If the antibodies are generated, it will get bound to the drug itself in the human body itself. So when we will measure these antibodies in assay, we will not be able to detect those antibodies. So it is very important to demonstrate the drug tolerance of your assay. So here you have to fix your quality control samples and you have to spike different amounts of drug in the quality control samples. And the point from which the drug starts showing positive results is your drug tolerance of the assay. Next slide, sir. Specificity. So you have to also demonstrate whether the particular antibody which you are detecting in the assay is specific to your biosimilar only or it is non-specific. So in this particular thing, you have to add another similar monoclonal antibodies or therapeutic antibodies in your sample, and then you have to detect it along with your target antibody, and you have to say that whether it is able to detect the antibody or it is not able to detect the antibody. So for ADA, different maps can be added, and it should be seen that whether it is able to detect or not. Next slide, sir. Again, selectivity is already explained by Dharmesh sir. It is nothing but the ability to detect analyte in a matrix such as serum or plasma that may have interfering substances. So again, in case of patient serum where there are more of RF factor and other interfering substances, it is required to demonstrate the selectivity of the assay. Also, sometimes the metrics are immobilized and lipemic. So in that case, also you have to demonstrate that your assay is able to detect the antibody in presence of such stringent matrices. Also, we are here. Also, we are doing stability as we are doing in PK assay, short-term stability and fixed stability as per the requirement of regulatory agencies. So this is all immunogenicity assays. Thank you very much.
Yeah. Thank you, Dharmesh. So these are little clippings of our recent inauguration of the laboratory. Thank you very much for giving us this opportunity, and I'll be happy if there are any questions, Dr. Somson. And thanks a lot for the attention of all the audience here.
Yeah, thanks a lot. Thank you, Dr. Ravi, for your excellent overview of various techniques which are you are doing in your industry related to the development of biosimilars up to clinical evaluation. And also, I thank the other scientists working in your institute, Dr. Dharmeshwar and Dr. Vishal, for explaining the procedure which what they are adopting for this pharmacokinetic study of biosimilars using ELISA as well as immunogenicity studies. I request audience if you have any questions.
I assume I probably we may be good here. So if there's anything, you can also send us through our email. And
Okay.
once again, I thank every one of you all.
Yeah. Thank you. Thanks a lot for your time and accepting our invitation.
Thank you. Thank you. Thank you.
Yeah. With this, we'll end today's program, and tomorrow morning, 10 o'clock, we'll meet with the next session. Thank you all.